Hello friends, in this lecture we'll talk about lipids. Here are some questions that have been asked in some of the CSIR examinations. On which among the following factors the fluidity of the membrane depends? Number 1, nature of head groups. Length of fatty acids is irrespective of the extent of unsaturation. Length and degree of unsaturation of fatty acid chains, none of the above. Another question was asked in June 2015. When bacteria growing at 20 degrees Celsius are warmed at 37 degrees Celsius, they are most likely to synthesize membrane lipids with more, and the options are given regarding the type of fatty acids that are present in the membrane. So for this we need to have the understanding about the types of fatty acids, saturated and unsaturated fatty acids, and their properties also. And so let's have a look at some of the basic aspects of lipids, and then we'll come to the discussion on these questions. Lipids. The lipids are a heterogeneous group of compounds including fats, oils, steroids, waxes, and the related compounds which are more related to each other by their physical rather than by their chemical properties. They have the common property of being relatively insoluble in water and soluble in non-polar solvents such as ether and chloroform. They are important dietary constituents not only because of their high energy value, but also because of the fat-soluble vitamins and the essential fatty acids which are contained in the fats of the natural foods. Fat is stored in adipose tissue, where it is also serves as a thermal insulator in the subcutaneous tissues and around certain organs. Non-polar lipids, they act as electrical insulators, allowing rapid propagation of depolarization waves along nerves. Combinations of lipid and proteins, which are called as lipoproteins, they are important cellular constituents occurring both in the cell membrane and in the mitochondria, and serving also as the means of transporting lipids in the blood. Let's have a look at the classification of lipids. Lipids can be broadly classified into simple lipids, complex lipids, and derived lipids. Simple lipids, these are the esters of fatty acids with various alcohols. These can be further classified into fats and waxes. Fats, these are esters of fatty acids with glycerol, such as oils, and these are the fats in the liquid state. Waxes, on the other hand, are the esters of fatty acids with higher molecular weight monohydric alcohols. Second class is that of complex lipids. These are the esters of fatty acids which contain, in addition to an alcohol and a fatty acid, they contain a more groups. So these further include phospholipids. These are the lipids that contain, in addition to fatty acid and an alcohol, a phosphoric acid residue. They frequently have nitrogen containing bases and other substances. For example, in case of glycerophospholipids, the alcohol is glycerol, and in case of sphingophospholipids, the alcohol is sphingosine. Glycolipids, these are the lipids which contain fatty acids, sphingosine, and a carbohydrate residue. Other complex lipids includes sulfolipids and aminolipids. Lipoproteins also may be placed in this category. Third class is that of precursor and derived lipids. These include fatty acids, glycerols, steroids, other alcohols, fatty aldehydes and ketone bodies, hydrocarbons, lipid-soluble vitamins, and hormones. Because they are uncharged, acylglycerols, also called as glycerides, cholesterol, and cholesterol esters, these are termed as neutral lipids. Here is the overview of the lipids which are found in the cells and tissues. Broadly, they can be of two types: storage lipids and membrane lipids, and some miscellaneous group is also there. So storage lipids, majorly they are consisted of the triacylglycerols, which have the glycerol in their structure, to which the fatty acids are esterified. In case of membrane lipids, the phospholipids, glycolipids, and archaeal ether lipids are included. Among the phospholipids, depending on the alcohol that is present, if it is glycerol, it is referred as glycerophospholipid. If the sphingosine is present, it it is called as sphingolipids. Similarly, in case of glycolipids, the sphingolipids are there and the galactolipids are there, which are also which can also be in form of sulfolipids. So here the sphingosine is present, which is esterified with the fatty acid, and the sugar is also present. Monosaccharide or oligosaccharide may be present in the structure. In case of galactolipids, the fatty acids are esterified with the glycerol, and mono or disaccharide may be present, which in turn may be connected with the sulfate. The sulfate may be present in such lipids. And in case of archaeal lipids, there is an ether linkage. So the glycerol and the fatty acids, they are linked to each other by ether linkage rather than by ester linkage. So this is the basic difference between the archaeal lipids and the other lipids which are found in case of bacteria or eukaryotic cells. Let's have a look at the fatty acids. Fatty acids, they occur mainly as esters in natural fats and oils, but do occur in the unesterified form as free fatty acids, a transport form found in the plasma. Fatty acids that occur in natural fats are usually straight chain derivatives containing an even number of carbon atoms. The chain may be saturated, that is, it may not contain the double bond, or unsaturated, which may contain one or more double bonds. So fatty acids, these are named after the corresponding hydrocarbons. So in the name of the hydrocarbon, the name ends with E. These are the normal alkanes. Now, in case of fatty acid, the corresponding fatty acid is, its name is replaced by OIC. Instead of E, the name ending in E, the name ends in oic. So if it is the corresponding alkane is octane, the saturated fatty acid is referred as octanoic acid. So in case of saturated acids, the enoic acid is the suffix, whereas in case of unsaturated fatty acids, the suffix is enoic acid. For example, octadecanoic acid is the unsaturated fatty acid, which is also referred as oleic acid. Carbon atoms are numbered from the carboxyl carbon. So carboxyl carbon is designated as carbon number 1, and the adjacent carbons to that of carboxyl carbon, they are numbered accordingly as carbon number 2, 3 and 4. Carbon number 2, 3 and 4. And these are also known as alpha, beta and gamma carbons respectively. The terminal methyl carbon is designated as omega carbon, and sometimes the numbering is also done from omega side. So omega 1, omega 2 and so on. That numbering also can be done. In case of unsaturated fatty acids, the double bonds may be represented by the number of carbons that are present in the chain of the unsaturated fatty acid, followed by a colon, and the figure indicating the number of double bonds, and after that a semicolon is used, which is then followed by a figure indicating the position of the double bond. So here the 18 colon 1 semicolon 9 indicates that it is the unsaturated fatty acid with 18 carbon long, and there is one single double bond in the structure, and the double bond is present between the 9th and 10th carbon. Often the delta sign is used to indicate the unsaturated fatty acid, and in the superscript the position of the double bond is mentioned. So delta superscript 9 indicates that this is an unsaturated fatty acid where the double bond is present at between the 9th and 10th carbon. Often the omega carbon is often used to indicate the position of the double bond. For example, omega 9 indicates a double bond on the 9th carbon counting from the omega side of the uh, unsaturated fatty acid. So counting from the omega side, the double bond is present between the ninth and the 10th carbon in case of oleic acid. As oleic acid is an 18 carbon long unsaturated fatty acid, if we look at the unsaturated fatty acid oleic acid, the from the carboxyl side also the double bond is present between the ninth and 10th carbon. So it is the case where omega numbering and the normal numbering is almost similar regarding the position of the double bonds. Saturated fatty acids. So here saturated fatty acids, they can be either short chain saturated fatty acids or long chain saturated fatty acids. So these are the short chain fatty acids that are shown. The number of carbon atoms that are present in acetic acid is 2, propionic acid has 3, and so on. These are the long chain fatty acids, saturated fatty acids, which are lauric acid having 12 carbons, myristic acid having 14 carbons, palmitic acid is having 16 carbons, stearic acid is having 18 carbons. So these palmitic acid and stearic acid, these are commonly found in the animal and plant fats. Lauric acid is generally found in coconut oils. Myristic acid is ample in coconut oils as well as in butter, nutmeg, and so on. Unsaturated fatty acids, they can be further divided as monounsaturated fatty acids if there is one single double bond, polyunsaturated fatty acids if they contain two or more double bonds, and icosanoids. These are the compounds which are derived from icosa or 20 carbon long polyenoic fatty acids, which comprises the prostanoids, leukotrienes, and lipoxygens. Prostanoids include prostaglandins, prostacyclines, and thromboxanes. So here is the list of several unsaturated fatty acids that are shown. Here is the list of the monounsaturated fatty acid like palmitoleic acid or oleic acid, which have only one double bond in their structure. It is palmitoleic acid is found in almost all the fats, and oleic acid is found uh, among the natural fats. It is most commonly found unsaturated fatty acid among the natural fats. Then allylic acid is there, which is present in the hydrogenated and ruminant fats. Uh, Among the polyunsaturated fatty acids containing the two double bonds, linoleic acid is the common one, which is found in the soybean and peanut or cottonseed oils. Then gamma linolenic acid and alpha linolenic acids, these are the uh, unsaturated fatty acids having three double bonds. These are found in some plant oils, and they are also found in the minor fatty acids in the animals. Alpha linolenic acid is frequently found with linoleic acid, but particularly in linseed oil. So these are some examples of unsaturated fatty acids. These are the hymnodonic acid is a polyunsaturated fatty acid having 5 double bonds. Carbonic acid is having 6 double bonds, and so on. So these are the polyunsaturated fatty acids which are found in the fish oils. So fish oils, cod liver oil, these contain hymnodinic acid. Carbonic acid is found in fish oils and phospholipids in brain. Here is the table indicating the uh, saturated and unsaturated fatty acids. So you can see these are the some of the saturated fatty acids which are found in several natural fats and oils, and these are some of the unsaturated fatty acids. Now you can see over here this table indicates the melting point. Now in case of saturated fatty acids, the melting points of these fatty acids are higher, and if we look at the melting points of the unsaturated fatty acids, the melting points are very lower. And that's why the oils which are rich in unsaturated fatty acids, they remain liquid in nature at even at room temperature, or even at lower temperature, they remain liquid because of the presence of polyunsaturated fatty acids. So these unsaturated fatty acids, as they have double bonds, they tend to have liquid state. So if the membrane is to be kept liquid, so if the cell is growing at lower temperature, it will increase its lipid content. Membrane lipids will have more unsaturated fatty acids in its structure. And as the organism is shifted to a higher temperature, then it may have some more content of the saturated fatty acid in its lipid content. So that is the main difference between the lipids that are found in the membranes, which maintain the fluidity of the of the membranes. So lipids, they can be storage lipids or structural lipids, which are structural lipids, they can be found in the membranes. So here is the figure indicating the overview of the lipids that are found in the cells, which we have already discussed. Triacylglycerols. Triacylglycerols are often referred as triglycerides, and these are the esters of glycerols and fatty acids. Mono and diacylglycerols, wherein one or two fatty acids are esterified with the glycerols, are found in the tissues, and these are particularly uh, they are of particular significance in the synthesis and hydrolysis of triacylglycerols. So here is the structure in which the glycerol esterified with the three fatty acids are shown, and you can see these are the ester bonds linking the fatty acids with the glycerol. These are the carbon numbers of the glycerol, SN1, SN2 and SN3. So these carbon numbers are different from each other, and that is evident from the action of several enzymes which act on glycerol. For example, the enzyme glycerol kinase, it will carry out the phosphorylation of the carbon number 3, SN3 carbon is phosphorylated rather than phosphorylating the SN1 carbon. So always by the action of glycerol kinase, the glycerol 3 phosphate is synthesized, not glycerol 1 phosphate is found. So that indicates that the carbon numbers in the glycerol are very much different, and these are numbered as SN1, SN2 and SN3, indicating the stereochemical numbering of the carbon. Phospholipids are the main lipid constituents of the membrane. Phospholipids may be regarded as derivatives of phosphatidic acid, in which the phosphate is esterified with the hydroxyl group of a suitable alcohol. The phosphatidic acid is important as an intermediate in the synthesis of triacylglycerols as well as the phosphoglycerols, but is found in any great quantity in the tissues. It is not much found in the tissues. So this is the phosphatidic acid, where the uh, uh, to a diacylglycerol at the third carbon, the phosphate is present. So this phosphatidic acid is generally found in association with the other compounds. Other groups are generally found attached to this phosphatidic acid. So phosphatidylcholine is the lipid in which the phosphatidic acid is connected to the choline. So this is the choline, which is having the three methyl group. Three methyl groups are present. So it is a labile carrier of the methyl group, and this phosphatidylcholine is it is generally found in the nervous tissues, and it is an important compound in the nervous transmission in form of acetylcholine, and it is a storage of the labile methyl groups. So here phosphatidylcholine contains the phosphatidic acid connected to the choline. Dipalmityl lecithin or dipalmityl lecithins are also referred as phosphatidylcholines. So dipalmityl lecithin will contain the palmitic acid at the two carbons, SN1 and SN2 carbons. So dipalmityl lecithin is a very effective surfactant agent and a major constituent of the surfactant, preventing adherence due to the surface tension of the inner surfaces of the lungs. Its absence from the lungs of the premature infants causes respiratory distress syndrome. Most phospholipids have a saturated acyl radical in the SN1 position, but an unsaturated radical in the SN2 position of the glycerol. Phosphatidylethanol amine, also called as cephalin, and phosphatidylserine, these differ from phosphatidylcholine only in that there is an ethanol amine or serine instead of choline in the structure. So here to the phosphatidic acid, ethanol amine may be present or the serine may be present instead of choline. Phosphatidylinositol is a precursor of second messengers. The inositol is present in the phosphatidylinositol as the stereoisomer myoinositol. So this myoinositol may be attached to the phosphatidic acid. This myoinositol may be present with the phosphatidic acid, and phosphatidylinositol 4,5-bisphosphate is an important constituent of cell membrane phospholipids. Upon stimulation by a suitable hormone, it is cleaved into diacylglycerol and inositol trisphosphate, both of which act as internal signals or second messengers. Cardiolipin is a major lipid of mitochondrial membrane. Phosphatidic acid is a precursor of phosphatidylglycerol, which in turn gives rise to cardiolipin. So here to the phosphatidic acid, if another phosphatidylglycerol is attached, it is called as cardiolipin or often referred as diphosphatidylglycerol. It is abundantly found in the mitochondrial membrane. Plasmalogens, these occur in brain and muscles. These compounds constitute as much as 10% of the phospholipids of brain and muscle. Structurally, the plasmalogens resemble phosphatidylethanolamine but possess an ether link on the SN1 carbon, instead of the ester link, which is found in the acyl glycerols. Typically, the alkyl radical is an unsaturated alcohol, and in some instances, the choline, serine, or inositol may be present instead of ethanolamine. So these are the plasmalogens. Sphingomyelins, these are found in the nervous system. Sphingomyelins are found in large quantities in brain and nerve tissues. Upon hydrolysis, the sphingomyelins yield a fatty acid, phosphoric acid, choline, and a complex amino alcohol, sphingosine. So sphingosine esterified with the fatty acid is called as ceramide. So this is the structure which is commonly found in the glycosphingolipids. Steroids, they play an important role in the physiology of the cells and the body. Cholesterol is probably the best known steroid. It is the precursor of a large number of equally important steroids that include the bile acids, adrenocortical hormones, sex hormones, vitamin D, cardiac glycosides, cytosterols of the plant kingdom, and some alkaloids. If we look at the structure of the steroids, they have these basic nucleus, steroid nucleus, where a cyclopentane ring is conjugated with the three ring structures A, B, C, and the methyl groups may be present on the carbon number on the position number 13 and 10, and there can be a hydroxyl group as in case of cholesterols. So hydroxyl group is present at the third position, and the carbon number 17 may be attached with a side chain. A side chain may be present at the carbon number 17 position. So this is the case of a cholesterol where a side chain is present on the 17th position and a hydroxyl group is present on the third carbon. And this hydroxyl group may in turn be esterified with the fatty acid, resulting in formation of cholesterol esters. So cholesterol is a significant constituent of many tissues. It is widely distributed in all cells of the body, but particularly in nervous tissue. It is a major constituent of the plasma membrane and of plasma lipoproteins. It is often found as cholesterol ester, wherein the hydroxyl group on the position 3 is esterified with a long chain fatty acid. It occurs in animals but not in plants. Glycolipids are the conjugated lipids in which the lipids are conjugated with the carbohydrates, and the carbohydrate portion it contributes to the cell surface carbohydrates. The major glycolipids found in animal tissues are glycosphingolipids. They contain ceramide and one or more sugars. Galactosyl ceramide is a major glycosphingolipids of brain and other nervous tissue found in relatively low amounts as well elsewhere. It contains a number of characteristic C24 fatty acids called as cerebronic acid. So here is the ceramide to which the galactose may be present attached. So galactosyl ceramide is there. In case of glucosyl ceramide, instead of galactose, glucose would be present. So galactosyl ceramide can be converted to sulfogalactosyl ceramide or called as sulfatide, which is abundantly found in the myelin, which is found in the myelin sheet, which insulates the neurons. Glucosyl ceramide is predominantly simple glycosphingolipid of the extraneural tissues, also occurring in the brain in small amounts. Gangliosides are complex glycosphingolipids which are derived from glucosyl ceramide that contain in addition one or more molecules of a sialic acid. Neuraminic acid is the principal sialic acid that is found in the human tissues. And gangliosides are also present in nervous tissues in high concentration. They appear to have receptor functions. Lipoproteins, these are the lipids which are conjugated with the proteins. These can be further classified into structural lipoproteins or transport lipoproteins. And structural lipoproteins, these are widely distributed in tissues, being present in cellular or subcellular membranes, in lung tissues and in eye. Transport lipoproteins, these are the forms which are present in the blood plasma. They are composed of a protein called apolipoprotein and different types of lipids, which include cholesterol esters, phospholipids, and triacylglycerides or triglycerides. As the lipid content increases, the density of plasma lipoprotein decreases. So these can be further classified into very low density lipoproteins, low density lipoproteins, high density lipoproteins. So very low density lipoproteins and low density lipoproteins, they have lower content of the proteins. And in case of high density lipoproteins, the protein content is more and the lipid content is less comparatively. And HDL, high density lipoproteins, these containing the cholesterol, they are referred as good cholesterol, whereas LDL containing the cholesterol, they are referred as bad cholesterol. Now these lipids, they have amphipathic nature, and because of that they can form various structures. So here a monolayer may result in the formation of micelle-like structure. For example, when the oil is mixed in water, the oil in water emulsion may form and the micelle-like structure may be formed. These lipids, they can also form a bilayer where the polar heads are oriented towards the aqueous phase and the nonpolar tails are oriented towards the inner side of the bilayer. And the liposome structure may be also formed where on the inner side the aqueous phase may be surrounded by the polar heads, and outside the liposome the aqueous phase is there, to which the polar heads are surrounded or they face the aqueous phase. So this is the liposome structure which can be formed upon the use of ultrasonicator. So ultrasonication can result in cavitation, resulting in formation of liposomes, and these liposome structures can be used for the transfer of drugs and so on. So let's have a look at the questions. On which among the following factors the fluidity of the membrane depends? Nature of head groups, length of fatty acids irrespective of the extent of unsaturation, length and degree of unsaturation of fatty acid chains, or none of the above. So here, as we have discussed, that as the length and degree of the unsaturation is differing, so the membrane fluidity will be affected. So here if the short chain fatty acid is there and the number of double bonds are more, then it can have more fluid nature. So length and the degree of unsaturation of fatty acid chains is very important for maintaining the fluidity of the membrane. Another question, when bacteria growing at 20 degree are warmed at 37 degrees Celsius, they are most likely to synthesize which kind of membrane lipids? So short chain saturated fatty acids, short chain unsaturated fatty acids, long chain fatty acids, saturated fatty acids, or long chain unsaturated fatty acids. So here you need to have the idea about the membrane fluidity affected by the length of the fatty acid and the double bonds, whether unsaturated fatty acid or saturated fatty acid. So unsaturated fatty acids will be required for inclusion in the membranes when the organism is growing at lower temperature. But when it is growing at higher temperature, then unsaturated fatty acids may be replaced with the saturated fatty acids. So here the long chain saturated fatty acids now would be incorporated into the membrane when the organism is shifted at the 37 degrees Celsius. So the correct option is the 3. So that was about lipids. Thank you and happy learning.